For over half a century, the foundation of neuroscience has rested on Hebb's postulate, the idea that neurons which fire together wire together. An isolated Hebbian model presents a specific biological problem. If every correlated firing event continuously strengthened synaptic connections, neural networks would reach memory saturation and succumb to runaway excitation. The human brain operates within the physical constraints of the cranium and draws from a limited pool of metabolic resources. Synaptic plasticity must exist as a regulated, bounded system. Models established in the mid-2020s define the brain as a strictly resource-bounded economy. Within this framework, learning is a competitive process where the stabilization of a circuit requires the active removal of subordinate connections. This is primarily achieved through heterosynaptic plasticity. When a neuron strengthens one specific input, it triggers a compensatory mechanism that drives the physical dismantling of adjacent, unstimulated connections on the same dendritic branch. The artificial network on the left lacks constraints, continuously adding glowing connections until reaching pathological runaway excitation. The biological model on the right utilizes heterosynaptic depression. When a target input strengthens, it forces the adjacent connection to retract, maintaining a stable baseline of excitation. The capacity to acquire information relies on the precise competitive elimination of structural noise. Historical neurobiology suggested that the synapse releasing the highest volume of neurotransmitters would inevitably win the developmental competition to wire into a circuit. In 2024 and 2025, researchers challenged this by examining the development of climbing fibers as they compete for space on cerebellar Purkinje cells. To isolate the role of chemical signaling, they utilized a targeted tetanus toxin light chain to completely ablate VAMP2 proteins, which suppresses all neurotransmitter release from specific climbing fibers. This diagram illustrates the competition. Under the old model, the active climbing fiber should easily secure the winner position, while the silenced fiber is pruned. The results showed that neurotransmitter-deficient fibers frequently bypassed the active fiber to secure the primary winner position. This indicates that initial selection is driven by intrinsic molecular identity and epigenetic markers, rather than electrical activity. The role of neurotransmission shifts once selection is complete. It provides the mechanical force required for the winning fiber to expand and translocate along the cell body. Functional synaptic transmission is the specific motor that executes the physical expansion of the winner and the heterosynaptic pruning of the losers. The brain establishes its structural hierarchy via pre-programmed chemical identity, but depends on electrical activity to execute the physical wiring of the circuit. To understand how a single neuron executes this choice between stabilization and elimination, we have to look at the chemistry within the dendritic spine. The synaptic tagging and capture hypothesis explains how multiple inputs compete for a highly limited pool of plasticity-related proteins trafficked from the cell body. When a synapse is strongly activated, glutamate binds to the NMDA receptor and creates a localized stabilization signal. This allows the specific spine to sequester plasticity-related proteins from the shared dendritic pool. As the primary input captures these resources, the resulting depolarization triggers a destructive molecular wave that propagates laterally down the dendrite toward adjacent connections. This lateral punishment signal is carried by Rho A, an intracellular molecule that regulates the reorganization of the cell's shape. Rho A physically dismantles the actin cytoskeleton, the structural scaffolding that gives a dendritic spine its physical integrity. Without its internal actin support, the unprotected, non-active spine rapidly collapses and is eliminated. Synaptic survival is a targeted process where the strongest inputs actively dismantle adjacent noise to prevent the neuron from being overwhelmed by divergent connections. Neuroscientists map these nanoscopic events into stable network behaviors using mathematical algorithms that govern how circuits maintain themselves over a human lifespan. Computational simulators like NEST and Arbor model this as homeostatic structural plasticity, where neurons physically grow or retract synaptic elements to maintain a target firing rate. This is the Gaussian growth rule.
It calculates the rate of synaptic change, dz over dt, by monitoring the intracellular calcium trace, C of t, as it oscillates between an upper boundary, epsilon, and a lower boundary, eta. The neuron uses its internal calcium concentration as a proxy for activity. If the concentration drifts outside these boundaries, the mathematical derivative shifts from positive to negative, dictating the creation or destruction of synaptic elements. When the firing rate is too high and the calcium concentration exceeds epsilon, the derivative becomes negative. The neuron actively breaks existing synapses to reduce incoming excitation. When activity drops and approaches the lower bound eta, the derivative turns positive. The neuron aggressively sprouts new synaptic elements to restore its baseline activity level. This model provides the logical bridge to information theory. Every synapse requires ATP to maintain resting potentials. By pruning inputs that fall outside these calcium bounds, the neuron eliminates the metabolic cost of maintaining low-efficiency, noisy connections that do not contribute to this signal-to-noise ratio. Structural plasticity serves as a mathematically precise optimization problem, maximizing the amount of information transmitted per single molecule of ATP expended. Local mathematical rules rely on systemic, body-wide executioners to translate chemical signals into physical changes in the brain's landscape. The neuroimmune axis provides this labor through microglia, the resident macrophages that constantly patrol the brain for underutilized connections. When a synapse lacks stabilization signals, the complement cascade deposits C1q and C3 proteins onto the spine membrane. This marks the specific synapse for recognition by microglial receptors. Microglia respond to these proteins through tragocytosis, actively engulfing and nibbling away at the marked synapse until it is permanently removed from the circuit. Endocrine regulation also plays a role. Surges in estrogen drive massive waves of hippocampal spinogenesis, resulting in fluctuations of synaptic density up to 30% across a mere 4-day cycle. Biophysical research from 2025 shows that these structural changes are further modulated by intrinsic circadian clocks. This chart tracks neuronal membrane capacitance. It oscillates on a robust 24-hour cycle rather than remaining static. Shifting capacitance forces the neuron to toggle its computational mode throughout the day. At the peak, it favors temporal summation of dispersed inputs. At the trough, it shifts to coincidence detection, requiring highly synchronized signals to fire. Memory consolidation and cognitive capacity are inextricably linked to peripheral immune surveillance, hormonal tides, and cellular biological clocks. This resource-bounded competition scales up to define the upper limits of human intelligence. Neuroimaging shows that a physically thinner cerebral cortex in the frontal and parietal lobes correlates with higher IQ scores in pediatric and adolescent groups. A thinner cortex is the physical result of highly efficient synaptic pruning. Eliminating weak connections reduces systemic noise and streamlines the metabolic cost of high-level cognitive control. Severe neuropsychiatric conditions emerge when the mathematical parameters governing this competition are perturbed. These neural network models demonstrate what happens when structural homeostasis fails. The hyperdense network on the left represents a failure of synaptic competition. In autism spectrum disorder, defective microglial signaling leads to underpruning, resulting in hyperconnected circuits that disrupt precise sensory processing. The right side illustrates the opposite extreme. Schizophrenia is driven by overactive competition and aggressive C4 gene variations that trigger excessive pruning during the critical adolescent epoch. The 2025 clinical frontier involves utilizing lipid nanoparticle mRNA therapies to bypass the blood-brain barrier and correct these pruning imbalances. Targeted delivery of brain-derived neurotropic factor can artificially restore stabilization signals to failing synapses. This provides a mechanism to halt the aberrant pruning cascades that define neurodegenerative conditions like Alzheimer's. Human cognitive capacity is the product of systematic structural elimination. The precision and stability of the mind are determined by the brain's ability to prune the subordinate connections of the neural landscape.